Okay, good afternoon, everyone. Today we are doing Beer's Law, the Beer's Law experiment. The objective of this experiment is to determine the concentration of an unknown solution by creating and using a Beer's Law plot from a solution of known concentration. Okay. So there are a few things we have to go over today. So first, let's talk about absorbance. So when every molecule on every molecule absorbs photons um, or electromagnetic waves in some manner. Some absorb in the UV, some absorb in the IR region, some absorb in the Raman region. But today we are concerning ourselves with the photons in the visible light spectrum. And if we use a spectrometer, we can actually measure this. So the amount of photons absorbed by a molecule is proportional to the number of molecules in solution, also known as concentration. So we can read the absorbance of a solution. And if we have a known concentration curve, we can figure out what the concentration of that solution is, what that solution is. So absorbance is proportional to concentration up to a certain limit. Okay. So when we are measuring absorbance, there are some other factors that we need to take into account. So the path length, or the distance that the light travels through a sample, is also important in reading absorbance. So for example, we have some images down here of two different lengths. So we have light passing through a large sample, in which a large amount of light starts in the beginning of the sample, and then as the light passes through, the molecules are absorbing it. So at the end, only a little bit of light is going through the sample. Now we have this skinny sample on the side here, and we can see that we start with the same amount of light, but it only passes through a short length. So not as much of the light is absorbed. And so when we are calculating concentration from absorbance, we need to take path length into account. So that means, like, how many molecules in that length does the light come into contact with? And how a spectrophotometer works is that it emits a certain amount of light, and the light passes through the sample, and then it detects how much light is left over at the end. And so the amount of light that goes through is how much light is transmitted through um, through the sample. Um, okay, so for example, the amount of light absorbed will be greater if the light passes through 1 foot versus 1 centimeter, things like that, even if it's the same concentration. Okay. Okay, so now let's talk about the last thing we need to take into account, and that is called the molar extinction coefficient. And this is a physical property of the material or molecule. So some molecules absorb certain wavelengths well, and some do not. And so we have this, this constant of a molar extinction coefficient that characterizes the material's capacity to absorb. And so the molar extinction coefficient is specific to a certain molecule. So the molar extinction coefficient of the Allura red we're using today would be different than the molar extinction coefficient of, say, bromethymol blue or something. So it is specific to the molecule. Okay. And so when we have all of these factors, so absorbance is proportional to concentration, but we also need to take into account molar extinction coefficient and path length. So, so now we have this equation: absorbance equals the molar extinction coefficient, which is this, this epsilon, times path length, which is B, times concentration. So this is Beer's law. And Beer's law we can plot. If we plot absorbance versus concentration, we get a linear relationship up to a certain concentration. And that concentration is going to be different for every molecule. But at some point, the spectrophotometer will not be able to read the transmittance or absorbance once you get up to a certain concentration, because the molecules are just blocking the light in addition to absorbing the light. And so it causes a kind of plateau when you get into high concentrations. But at the lower concentrations, there's a clear linear relationship between absorbance and concentration. And so today we are going to be creating a plot just like this from our data. Okay, so in the in the experiment today, we will be using a micropipette. And micropipettes have pipette tips. So in the micropipette, we can have a specific volume that we want to dispense. And so Amy in the video will show you how to do that today. Okay, so in the experiment, we're using a spectrophotometer. So because we are using a solution that is red, the solution looks red because it is absorbing green light. So because we know that our solution is absorbing green light, we want to make sure that we set the wavelength to a wavelength that is in the green area of the visible spectrum. So we are going to be using the wavelength 520 nanometers. And the first thing we want to do is make sure that the spectrophotometer has time to warm up its little light bulb so that we can get some accurate readings. Okay, so the other thing we are doing today is we are doing some serial dilutions. So we need to prepare the solutions from a known concentration from a stock solution. You will see in the video that Amy uses a 2 millimolar solution. However, on the data that was given to you guys, the first standard is 1 millimolar. So when we do some sample calculations, that's what we will be using as our standard. So what we would do was we would record the concentration from the bottle, and then we would make 8 serial dilutions for a total of 9 solutions. So the first solution would be our standard, and then we would have 8 dilutions. So this is just an example of how to make a serial dilution. This is not the dilution we're doing today, so I'm not going to go through it right now. Okay. So when we prepare the spectrophotometer, once the lamp is warm, it just takes a few minutes. We first need to blank our spectrophotometer. So what that means is we will fill the cuvette with deionized water. And we want to make sure that that the sample is filled in between this little triangle and this little rectangle here. And we would wipe the cuvette down to make sure that there's no lint, there's no dust, no fingerprints, and we would place that cuvette in the spectrophotometer and take our absorbance reading. And those absorbencies are going to be used to find the lower limit of detection. And so Amy will go through that in the video. And then once we've done that, we use that same DI water and we can press the 0 absorbance button or the 0 button. Sometimes it just says the word 0 to blank the machine. And so that that basically just takes the background of the machine so that it knows that any reading due to the water, it will subtract that from the absorbance. Okay, and then we need to start reading the absorptions, and we're starting with the most dilute sample. So we'll add that and start reading the absorbencies. The really dilute samples oftentimes is indistinguishable from water, and we'll see that in the video. Okay, so here's an example of the bromophenol blue Beer's law plot. Again, this is not the molecule we're using today, but this is an example of what a Beer's plot would look like. So the lower readings are at about 0. And then as we get more and more concentrated, we have this linear area, and then they kind of plateau off at the higher concentration. So on the Beer's law plot, we are going to be finding what the equation of the line on the linear portion is in order to find the absorbance. Okay, so we will be plotting today in Excel. So hopefully you all have Excel. Okay, the last thing we're going to do is we are going to determine the unknown concentration. So there's going to be some unknown solution, and we are going to measure that absorbance. So we are going to dilute it and then measure the absorbance, I'm sorry. And so then we can figure out what the dilute concentration is. And then using M1V1, we can figure out what the starting concentration was. Okay. Safety, gloves and goggles. In this one, all the waste can go down the sink. So today we just have the data sheet, so you won't be handing in a report, just filling out the data. So you need to calculate the concentrations of the diluted standards, the Beer's law plot, calculate the molar absorbent or the extinction coefficient of Allura Red, estimate the upper and lower limits of quantification, use the absorbance of the diluted solution to figure out the unknown, etc. So there are questions at the end. I believe these are also in your... Okay, so if you can see on the screen, you need to do everything that the data sheet says, and then there's this little discussion section. So you have to answer these questions and hand those in as well. Okay, so let's watch Amy's video and then we can start doing some of the things together.
All right, so the first thing I've done is I've turned on my spectrophotometer and set the wavelength to 520 nanometers, which is the wavelength we'll be using to measure the absorbance today. So that's done. I had to let it warm up for a minute, so I went ahead and did this ahead of time.
Sorry, I forgot I had it in 2 times.
All right, so the first thing I've done is I've turned on my spectrophotometer and set the wavelength to 520 nanometers, which is the wavelength we will be using to measure the absorbance today. So that's done. I had to let it warm up for a minute, so I went ahead and did this ahead of time. The next thing that I'm going to do is measure the lower limit of quantification, I believe. Yes. So we're going to do this by measuring the absorbance of a clean cuvette with DI water. So first I'm going to rinse my cuvette several times with DI water. To make sure there's no residue in there. Just making sure to get the sides. I'm then going to go ahead and fill it with some DI water. Making sure that I have enough liquid in there that when I put it in the spectrophotometer, it will be at a measurable level. So I'm going to wipe it off with a Kim wipe, make sure it's completely clean, there's no smudges on the outside. So now that we have the cuvette, we can go ahead and take the measurement. I've cleaned it off. I'm going to gently insert it into the spectrophotometer. I'm not going to jam it in there or force it. You will feel where it stops. I'm going to close the lid and write down the measurement seen here. A is for absorbance, so that's at 0.049. I'm now going to take this measurement 10 times to determine how well our spectrophotometer is operating. All right, I'm now going to make our dilutions. We're going to have a set of 9 dilutions. I'm going to start with the DI water. So I'm adding 1,500 microliters to each of my test tubes here, except for my standard. So I'm going to add the water first. And 1,500 microliters is the same as 1.5 milliliters. All right, so the instrument I'm using here is a micropipette. It reads in microliters, which is why I went ahead and converted the milliliters to microliters. And in just a second, we will zoom in on where you can see that measurement. But when you're using a micropipette, you're going to push down to the first stop to draw liquid up into the pipette. And then when you're dispensing it, you are going to push down to the first stop and push to the second stop in order to dispense all of that liquid. You want to make sure that you never tip this micropipette upside down when it has liquid in the tip. This will ruin the micropipette because it will get the mechanism essentially clogged with liquid, and that's going to cause problems. So if you take a look at the side of the pipette, you can see that's reading at 1,000. It's 1,000 microliters or 1 milliliter. These pipettes do come in different amounts or different ranges. This one will measure up to 1,000 microliters. Some only measure up to 200 microliters. You'd use the appropriate one for what you were doing. For today, we're going to use the 1,000 microliter pipette. I'm now going to adjust it by twisting the end. You also want to make sure that you never twist it above the amount that the micropipette is capable of measuring. This could also cause damage inside the instrument and cause it to read less accurately. So we're now going to move it down to 500 microliters. And add the 0.5 milliliters of water to all of my test tubes. All right, so now I've gotten a clean pipette tip. I'm now going to obtain 2 milliliters of my standard. As you notice, that is a bright red solution. You want to make sure that you write down the starting concentration so that when you do your dilution calculations, you know what concentration to start with. And that's always going to be written on your reagent bottle. I'm now going to go back to my workstation and add, I'm adding 0.5 milliliters of my standard solution to the first test tube. I will then add 0.5 milliliters of this solution that I'm going to create into my next test tube, and this is known as a serial dilution. So I'm now going to add 500 microliters of my standard solution to my next test tube. For the sake of mixing, I'm also going to draw some of the liquid up and dispense it back in. Make sure this is thoroughly mixed. I'm then going to discard my pipette tip and get a new one for my next dilution. All right, I'm now going to take 500 microliters of my freshly made solution. I'm going to add it to my third test tube, mixing it. And so on and so forth. I'm going to make 9 of these dilutions. And this is what's known as a serial dilution, because I'm taking one solution, making a new one, then using the new solution to make a third, so on and so forth. All right, so I'm now going to insert my cuvette with my DI water, close the lid, and I'm going to press the 0 absorbance button, and that's going to blank our instrument, essentially telling it to negate the absorbance that it's reading for the DI water. So that won't affect our future measurements. So I'm now going to measure the absorbance of each of my diluted samples. I'm going to start with the most dilute so that I don't contaminate the cuvette with essentially staining it red or additional red drops. So if you note, this solution is almost clear, where my standard solution is this bright red color. So we don't want any of this getting in the more dilute samples. So we always go from most dilute to most concentrated when we're measuring absorbance. I'm going to take enough solution out of here and place it in my cuvette. And I do have a few water droplets in my cuvette, which will dilute it slightly further. So that would be a source of error. Realistically, you would want to start with a dry, clean cuvette. So I'm now going to wipe my cuvette off to make sure there's no fingerprints or smudges on it, or water droplets for that matter. I'm going to insert it into my spectrophotometer and measure the absorbance. All right, so before I do my next solution, I'm going to dispose of my liquid in the waste beaker. I'm going to rinse out my cuvette with DI water to make sure there's no residual solution in here. And I'm now going to add my next solution after I increase the amount of liquid that my pipette's going to draw up. All right, I'm now going to take my second least concentrated solution and fill the cuvette. And we are then going to record the absorbance. Now, so you notice this absorbance is reading at 0.000, which means that it cannot differentiate between this sample and the DI water. All right, so I'm now moving on to the second part of the lab where we're going to make a 1 to 10 dilution of a solution with an unknown concentration of the same red dye that we just tested in our standards. So I've made the calculation of how much water I need to dilute 9 parts water to 1 part unknown solution. And I'm simply going to use M1V1 equals M2V2 to make that calculation. So I've gone ahead and done that for the sake of time. And I know I need 3.6 milliliters of water for 0.4 milliliters of my unknown solution. And I'm doing this in a beaker because the amount needed would not fit in these small test tubes. So to make sure that I don't overflow them, I've gone ahead and made it in a beaker. Now I'm going to mix that to make sure it's nice and mixed. And now I'm going to add a small amount to our cuvette. Something to note is you want to rinse the cuvette with a small amount of your solution, making sure you get the sides wet prior to putting the solution into your cuvette to test it in the spectrophotometer. Now I'm just going to measure the absorbance the same way that I measured the standards and record that absorbance value. Should be good. And that absorbance value is 0.233, so I'm going to write that down. And that is it for the experimental section. We'll then go into some of how to process the data. All right, so just a few things to help you guys with your calculations for this lab. Hopefully you're familiar with M1V1 equals M2V2, but just in case you aren't, we're going to go over that quickly. M1 stands for the concentration of your first solution. V1 stands for the volume of your first solution. So if you know the volume of the solution that you're trying to create, which would be V2, or the concentration that you're trying to create, M2, you can plug those numbers in and solve for your unknown. This calculation is great for dilutions because you will know the starting concentration, M1, the starting volume, V1, and you will know your V2. You're just going to be looking for your M2 value. So, taking a look at this board, this is how you are going to calculate the concentrations of your dilutions that we did in the lab. You will start with your starting concentration of your standard, which is 2 millimolar. If you're using millimolar, you need to use milliliters. If you're using molar, you would use liters. So that's interchangeable when you're doing these calculations. Just make sure you don't mix and match. So we started with a concentration of 2 millimolar of the dye. We added 0.5 milliliters to form a total solution of 2 milliliters of dye and water. And after we perform the math of this calculation, we find out that our concentration of our first dilution is 0.5 millimolar. This value is now going to be your starting concentration in your next dilution. So when you go on to do the concentration of solution 2, you need to start with the concentration of the first dilution, not the original standard. You will go ahead and repeat that calculation for all 9 dilutions, and that's where those concentration numbers are coming from. You're also going to plot a graph of absorbance versus the concentration that we evaluated in lab, or that we measured in lab. So you're going to get a curve that looks something like this. You should have a straight line section. I apologize, my drawing is not the straightest. But you also find a section on your graph where you start to see a curve. You want to look primarily at the linear section of your graph. Your y-intercept also should ideally be at 0. So for this lab, you're going to use this graph to determine the concentration of your unknown solution. So remember, we took an unknown concentration of the dye, we diluted it in a 1 to 10 dilution, and then we found that absorbance. So we're going to use the absorbance of the unknown and determine the concentration from this graph. How you're going to do that is you will find the equation of your line using Excel, and it's going to give you the typical equation of a line, y equals mx plus b. Y is going to be your absorbance, and x is what you are solving for. B is your y-intercept, and m is the slope. So this lab also wants you to calculate the molar absorptivity. You might be wondering what that is. If you ever see the equation A equals EBC, the Greek letter epsilon is what stands for the molar absorptivity. A is the absorbance, B is the path length, and C is the concentration in molar. The path length is in centimeters. And it's important to make sure that you're in the correct units so that you don't get these numbers mixed up incorrectly. So, in order to solve for your molar absorptivity, we've given you that the path length is 1 centimeter. You'll plug in concentration and the absorbance and solve for your molar absorptivity. And that should be enough to get you going with these calculations. If you have any questions, feel free to ask your TA.
Alrighty. So let's let's go through the Beer's Law stuff. All right, so what do we need to fill out? So we need to do the lower limit of quantification. So let's take a look at the makeup data. So one thing I want to say about the makeup data, this first concentration says 1 times 10 to the minus third. That should be 2 times 10 to the minus third. Okay, so in your data, when you're copying this data sheet down for table 2, make sure that you change this from 1 to 2, okay? All right, so our lower limit of quantification data is this here. So we've given you all the readings. The only thing you need to do is find the average absorbance. So take the average of these values and find the standard deviation. And the lower limit of quantification should be that average, I believe. Hold on one sec. Okay, so when you multiply the standard deviation by 10, that is your lower limit of quantification. So when you find the standard deviation of this data, multiply it by 10, and that's this here. Okay, and so we've given you this data. I realize that it is much more than you need, but we can easily just plot this all in Excel. Okay, so let's go through what you need to hand in. So we have Beer's Law here. Okay, so let's do the calculations for the dilutions. So I know that they give you all of the concentrations, but this is something that you will need anyway. So for our standard one, so our first standard, the concentration is 2 times 10 to the minus third molar, or 2 millimolar. Okay, so we're going to keep everything in molar because the data that was given to you was in molar, so that's fine. So we need to find standard number 2. So to do that, we need to know a few other things. So the volume of standard number 1 that we're using or transferring is 0.5 milliliters. So we're taking 0.5 milliliters of our 2 millimolar solution, and to make a 2 milliliter solution. So what is M2? So if we put this into M1V1 equals M2V2, we have 2 times 10 to the minus third molar times 0.5 milliliters equals M2 times 2 mils. So we're going to divide both sides by 2 mils. So this cancels out and our milliliters cancels out. So what we have is oops. Our M2 equals 2 times 10 to the minus third molar times 0.5 divided by 2. And that equals 5 times 10 to the minus fourth molar. Okay, so this is the standard number 2 concentration. So now we need to make standard number 3. So standard number 3 is going to be equal to M2 times 0.5 mils over 2 mils, because we need to make a 2 milliliter solution of M3 using 0.5 mils. So this is the same exact thing, except for we are substituting M2 into our equation. So M3 is going to be equal to 5 times 10 to the minus fourth molar times 0.5 divided by 2. And this is going to give us 2.5 times 10 to the minus 4 molar. Right. I'm going to double check that. Yes. Okay, and so for standard number 4, 4 would be equal to M3 times 0.5 over 2. And that's going to be whatever. So we already have that in our data. So that would be 1.25 times 10 to the minus 4 and so on. And we would do that until we got 9 total samples. Okay, so, and then the next step would be to read the absorbance of all those guys. And so in your Beer's Law data, we would put that here. So the concentration of the dye and the absorbance. So we would start with the 2 molar and go down until we have 9 of them. Okay. So does anybody have any questions on the M1V1 calculations?
No, but I just have a question. Are we supposed to use all the data? Because on the table it says just 9.
So you can just use 9 of them.
Okay.
So let's, yeah, you're welcome. So let's do that together now. So I'm gonna bring up Excel. So everybody, if you want to bring up your own Excel, we can do this together. Okay, so. Okay, so we need to do concentration versus absorbance. So in Excel, when you are plotting data, the column on the left is usually going to be the x-axis and the column on the right will be the y-axis. So let's go ahead and copy in that data to Excel. And I can show you how to make the plot. So the E that you see there stands for 10 to the power of. Okay, so let's copy that. So 1e minus 3. And I even forgot the first one. Remember that it's a 2, not a 1. Okay, and then copy down the absorbencies. So 3.1, 2. Okay. So now we have our first 9 readings. So again, make sure this is 2 times 10 to the minus third, or 2e minus 3, or 0.002. Okay, so we are going to select our data. Then insert, and we want a scatter plot. So we want the little dots here. Okay. So now I'm gonna make this bigger so we can see our linear portion. So the linear portion of our plot is going to be over here. So now we want to add a trend line. But we only want to add it for part of the data. So we can So we are going to want to plot the data, only the data that we see that is linear. So we can choose, let's see, I believe that this data point, so 2.5 at 2.5 or 0.25 millimolar, that one doesn't really look linear. So the data, so we can delete these 3 data points because they are not linear, so we don't need them. So if we go back to, if we click on our data, we can go back to our data here and modify our data. Come on. No. Okay. Let's just add a new plot. So because we know this data point here is no longer linear, and so are these guys. These guys are plateauing. Let's select this data and make another scatter plot. Okay, so this data is linear. So now we want to go to this, we want to select the chart area. We want to add a trend line, and it needs to be linear. Okay. So let's go to format trend line. So in format trend line, we can display the equation on the chart, and we want to also display the R-squared value. So the R-squared value tells us how close our data is overall to that trend line. So the closer to 1 that we get, the better. Okay, so here is our data. All right, trend line. Okay. So we want to also set the intercept to 0. So at 0 concentration, there should be a 0 absorbance. All right. So now we have our equation of our line. And so the equation of the line is what we are going to use to find our unknown. Okay, so let's go back to this guy. All right, so what have we done so far? So we plotted our data. We determined the slope of the linear part of the line. We have the equation and the R-square value. So calculate the molar absorptivity of the Allura red in water, given that the path length of your cuvette was 0.1 centimeters. So for this, we can choose one of our linear data points. Let's see. So we can choose one of our linear data points. So let's choose one in the middle. So let's use 3.13 times 10 to the minus fifth. We'll use that data. And let's go back to our whiteboard. And so now we are going to be using A equals epsilon B times concentration. So we're going to use the concentration at 3.13 times 10 to the minus fifth molar. And we're using this concentration because we know it's in the linear portion of our graph. So we know that we can directly, we know that this concentration will be proportional to absorbance. We know for this concentration, the absorbance was 0.679. And path length path length B is 1 centimeter. So now we just need to find epsilon. So let's plug these in. So 0.679 equals epsilon times 1 centimeter times 3.13 times 10 to the minus fifth molar. And you might be asking for path length, how do we know it's 1 centimeter? The standard size of every cuvette for all spectrophotometers is 1 centimeter. So that's what we always use. So now we have to solve for epsilon. So 0.679 divided by 3.13 times 10 to the minus 5th molar centimeter equals epsilon. And so now we just have to do that calculation. So 0.679 divided by 0.1234313. And so our molar extinction coefficient is 21693. So let's take a look at the equation of our line. That's pretty close to what we got for a slope. So that pretty much checks out. Okay, so does that make sense to everybody how we did that? All right, so let's see what else we have to do on this page. Okay, estimate the upper and lower limits of quantification. So the lower limit is the standard deviation. So we'll do that in a second. So the upper limit on here is going to be our most concentrated. So our upper limit in this case would be 1.25 times 10 to the minus 4th molar, because that is our last linear data point. So as far as we know, based on the serial dilutions that we did, that is as linear as we got. So that is our upper limit. Okay. All right, so let's do the lower limits of quantification now. So I'm going to add another sheet and let's do that data. So I will show you how to get the standard deviation in Excel. All right, so, oh, I need the sample data. Okay, so let's just put those in. So 0.003. 0.001, negative 0.040, 0, negative 0.002, 0.002, 0.005, 0.001, 0.003, negative 0.002. Okay, so to find the standard deviation of these guys, we can use Excel directly to do this for us. So the first thing we're going to do, we also need this average of the absorbance. So we can do equals and then type in average of, and we need to tell it which cells. So we need to do cells A1 through A10. Okay, and so here is our average. I'm gonna insert. So this is our average. And then now we need our standard deviation. So for standard deviation, let's start typing that in. So standard dev, it gives us a lot of different types of standard deviations. So standard deviation.p is the standard deviation of a population. So if you're doing like population biology, that's what you would use. But we just have a sample. So we want to use standard deviation.s. Okay, so that's the standard deviation we're going to use. And again, you can select B1 and then shift to B10. Okay, so now we've used Excel to do the standard deviation, and the lower limit of quantification is your standard deviation times 10. So we can just do equals 10 times, and then select our standard deviation cell and press enter. So our lower limit of quantification is 0.027508. So now we have that done. So let's see what else we need to do. Okay, so use the absorbance of your diluted solution, the slope and intercept of your Beer's law plot, and your dilution factor to calculate and report the concentration of Allura Red. Okay, so let's do that. So let's go back to our plot. And let's take a look at our makeup data. So let's go down to part 2. So we used a volume of unknown solution and water to make a diluted solution. So we need to use Beer's law again. Okay, so let's first write down y equals mx plus b. So we set the y-intercept to 0, so we don't need to use that portion. So our equation of our line is y equals 18531x. So on our chart, our y-axis is absorbance and our x-axis is concentration. So what that means is A equals 18531 concentration. So in our diluted solution, our absorbance was 0.565. So 0.565 equals 18531 C. So C equals 0.565 divided by 18531. So this is equal 3.05 times 4 times 10 to the minus 5 molar. Is everybody with me so far? So this is the concentration of the dye in the diluted solution. So in order to make this dilution, we had M1 and we took 0.4 mils of it to make 3.05 times 10 to the minus 5 for a total of a 4 milliliter solution. Does that make sense so far?
Is it 0.4 ml?
So we took 0.4 ml of the original solution to make a 4 milliliter solution that we just figured out has a concentration of 3.05 times 10 to the minus 5 molar. So on the makeup data sheet, it says volume of H2O added to make the diluted solution. It's not 2 milliliters. It should be 3.6 milliliters. I don't know why they have that there, because where is it? Here. So in line 13, it says measure about 0.5 milliliters into a beaker and add 9 times as much to create a 1 to 10. So 9 times 0.4 is 3.6. So 3.6 would be the volume of H2O added. So that is an incorrect number there. Okay, so that total 0.4 plus 3.6 milliliters is a total volume of 4 milliliters. Okay, so now we need to solve for M1. So M1 equals 3.05 times 10 to the minus 5th molar times 4 milliliters over 0.4 milliliters. And so our M1 is going to be equal 3.5 times 10 to the minus 4th molar. So that is going to be the concentration of the dye in the original juice. Okay, so let's take a look at the data sheet again. So, so now we've done part 1, we've done part 2, and part 3. And for the data part, we've done the prepares the plot, we've calculated the molar absorptivity, we have the upper and lower limits of quantification. We figured out what the original concentration was. So now all you guys need to do is the discussion. So comment on the quality of your Beer's Law plot. So, and it says how linear is your plot, and what does the y-intercept say about your plot? So let's take a look at our trend lines. So we did set the y-intercept equal to 0. Oops. So. So when we don't set our y-intercept equal to 0, we have a 0.062. So how linear is your plot? The R-square value tells you that. So the closer to 1 you are, the closer you are to being perfectly linear. So you can discuss this R-square value. In Beer's Law, we don't have a plus B, right? So the intercept should be 0. So think about that as well. So our y-intercept is a little bit off of 0. So talk about that as well. Amy does discuss some of the sources of error in the experiment. Now remember, when you're talking about error, calculation is not an error. So you can, if you make an error while you're calculating, you can always recalculate. So you need to think about the things that are behind the calculation. So our calculations are about concentration and about absorbance. So you want to think about things that we did experimentally or that Amy did in the procedure that could have affected the absorbance readings or the concentration. So think about those sources of error. And then finally, provide a summary of your results. So basically discuss the results. So what that means is, were you able to successfully use Beer's law to find an unknown concentration? Because remember, that was the purpose of today's experiment. So talk about that. Now, does anybody have any questions?
Yes, I have a question.
Yeah.
So for the graph, because I see you did some like data points. So are we supposed to do the same, or like, are we supposed to like make two graph, or just the graph that you made that was linear, one specific part that is linear, then delete the data point that aren't linear?
Right. So that's a good question. So if you know how to just do a line on part of a graph, you can keep the whole things. I don't know
Okay.
how to do that.
Okay.
So you can, the other option is just to create the plot with only the linear data points.
Okay.
So
Okay.
you can include both things in your submission if you want.
Okay, I won't get penalized for that.
No.
Okay. Thank you.
Yep. Okay, does anybody else have any other questions?
When do we have to actually submit the data sheet with all the questions?
So today is Tuesday, so your submission date is,
I think it said Friday on
I
Blackboard.
believe it's Friday at midnight.
Oh, okay, thank you.
Yep.
For the upper limit of quantification, how did you find that again?
So the upper limit, based on what we know, is that if we take a look at our plot of all the absorbance data, the linear portion, we can only go up to this data point here. So after that, all the other data points are no longer linear. So what that tells us is that the machine can at least read up to this concentration of this data point, but everything else is does not follow Beer's law. So that means the upper limit of quantification for us is this concentration. So if we had more dilutions in between these two data points, we could get something more specific, but we don't. So this is the most accurate we can get for our highest concentration.
Okay, thank you.
Yep. Any other questions?
Can you just repeat what you said for the first discussion question about the linear?
Yeah.
How you said the Beer's law is close, the y-intercept is closer to 0.
Yes. So when you have 0 molecules in solution or a concentration of 0, then the absorbance should also be 0 because there aren't any molecules to absorb any of the light. So because we have a positive y-intercept, that means that our readings are a little bit off because it's causing the equation of the line to have if we substitute 0 in for x, then our y is going to equal a positive value. So if our concentration is 0, there shouldn't be any absorbance.
Okay.
So that tells us our readings are a little bit off. And then similarly, the R-square value tells you overall how close your data points are to fitting the line. So an R-square value that's equal to 1 is perfect. So the closer you are to 1, the better your data is. So we can see that our data is not quite perfect based on our R-squared value, but it's very close.
Okay, that makes sense. Thank you.
Yep. All right, any other questions? All right, so that's all I have for you guys today. So I'm gonna stop sharing and I'm gonna stop recording. Oops, wrong thing.